Good morning, everyone. Let's get started. Okay, so I think this was the place where we left last time. So today we're going to continue to talk about the interaction between different alleles for a single gene. So pay attention. Right now we talk about just one gene. But I think this gene, one gene can take different forms, different alleles, right? So the relationship between different alleles would be like the dominance or recessive. So this is what we're gonna start talking about today. All right. So so here we talk about like, for example, a wild type allele and a mutant allele. So the big A, the wild type allele, is dominant in related to the mutant alleles, lowercase a, right? So this is a very important concept, the haplosufficient. So what does this mean? So we have talked about this concept before, right? This means like, so in a diploid individual, we have 2 copies of this gene. Actually, in an individual, only a single copy of the wild type is sufficient. So that's if this is the case, we say this is the haplosufficient. Look at here. So for this individual, this is the heterozygous. There's only 1 copy of the wild type allele, but this one copy is good enough. It can produce enough protein or enzyme that can lead to the wild type. So in this case, for the haplosufficient, the homozygous big A will show the same phenotype as the heterozygous. They're showing the same wild type. Only if an individual carry like 2 mutant alleles, then this will show different phenotype. So haplosufficient means the homozygous wild type, homozygous dominant alleles will have the same phenotype as the heterozygous. All right, now it's easy for us to understand haploinsufficient, right? Haploinsufficient means like, all right, so in the heterozygous individuals, so only 1 copy of the allele, wild type allele, is not enough. So for example, this is a very simple model to for understanding the biology behind this. So suppose the C2 allele, this is 1 gene. So we have 2 C2 alleles, 2 wild type, and each of this C2 allele can produce some protein, functional protein. Now there are 2 doses in this individual, right? Now, when we look at this heterozygous individual, there's only 1 copy of the C2 allele, wild type allele. The other one is C1. So C1 is mutant. It cannot produce any protein. So this single C2 allele can produce 1 dose of the protein. But this 1 dose cannot lead to the same phenotype like this 2 dose. That's why you're gonna see like for the wild type, you're gonna see darker for the darker color in the flower petal. If you look at the heterozygous, you're gonna give you it will give you some color, but not as dark as the homozygous, right? All right. So only so if both alleles are mutants, it will not give you any color. So that's the white. All right. So this sometimes we call this incomplete dominance, or sometimes like some people say it's like incomplete dominance is kind of like equivalent to the codominance because both alleles are expressed in this situation. So I hope like this can help us to understand the difference between haplosufficient versus haploinsufficient. Okay. Okay, so this is the model, the gradient model that can help us to understand the incomplete dominance. So for example, the flower color in this case. So the mutant, the homozygous mutant C1C1 allele, it can produce 0 enzyme that can produce the purple pigment. All right, so this is the 0. So now let's look at the heterozygous. So only one functional allele, wild type allele, it can produce this much enzyme. And so when we compare the homozygous wild type to this two, so it can produce like doubled, right? When we compare to the heterozygous, the homozygous wild type can produce doubled amount of the enzyme. Of course, so the flower will be darker because of a significant more proteins or enzyme can be produced in this individual. So this is one model, the gradient model can explain the incomplete dominance. So for the complete dominance, so the complete dominance, there's a model. Also, there's a model, threshold model, which can be used to help us understand the biology behind. So, for example, the wild type. Oh, okay. So, for example, in this case, a normal phenotype will require 8 units of some enzyme, 8 units. So for example, this is 8. The y-axis is the unit. The y-axis is unit. So the 8 is the threshold. So if an individual can produce the enzyme more than 8 units, then it will show the black phenotype. Otherwise, if an individual cannot produce more than 8 units of the enzyme, so it will show colorless, something like this. Right. Okay. Now, so each normal functional allele can produce 10 units, which means like the big A can produce 20 units, like here. Big A. So of course, in this big A individual, so it can produce more than 8 units. So it will show the wild type phenotype dark, right? So now, if we look at the heterozygous, this individual can also can produce 10 units, which is also greater than 8. So then this is why you also see the wild type phenotype dark. So this is complete dominant. However, if you look at the homozygous recessive alleles, this can produce 0. So yeah, it is lower than 8. So then it will give you a different phenotype. So in this case, we say big A is complete dominant to little a. So this is a complete dominance situation. Threshold model. Okay, now let's move to the third type, the lethal allele. Lethal alleles. Okay, so any alleles that can result in death. So this will call the lethal allele. So either this or this or this can be lethal. So in the nature, but usually like the lethal alleles, the most lethal alleles are in this form. So the recessive lethal. Okay. So for example, there are some examples in human disease, like we talked about this before, right? Achondroplasia syndrome, or the PKU. So they are all recessive lethal. So we're going to talk about this shortly, right? So how do we? So how can we identify this lethal alleles? Usually, so if we look at the population, or if we look at a Punnett squirrel, so the proportions of the progeny from the cross, or in the population, if some phenotypes show showed unequal distribution in the population. So this will give you a strong indication that probably, yeah, this phenotype is associated with some lethal alleles for a gene, all right? And usually, when we talk about the lethal alleles or lethal genes, it shows the pleotropy phenomenon. So this is another term, so important term here. So the pleotropy means any allele that can affect multiple phenotypes. So a allele for a gene, so if that allele can produce more than one phenotype, so then we say this gene or this allele is a pleotropy. Okay. All right, let's look at an example, the lethal alleles here. Right. Okay, so this is an example in mice. All right, so there's one gene, Y gene. Okay, so when we look at this Y gene, so the big Y and the small y, so first this gene is a pleotropy gene, so which means this gene can produce more than one phenotypes. So in this case, so this gene, so first this gene can determine the color of the coat for the mice. So the big Y, so in terms of this phenotype, the color, the coat color, so the big Y, capital Y, is the dominant. This will give a mouse a yellow coat color. All right. So that's why this one is yellow, and this one should be yellow, and this one should be black. So homozygous little y will give a black coat for a mouse. However, the same gene, this can lead to another phenotype. This can determine another phenotype, which is the being alive or not. So as you can see here, so if we just look at the first phenotype, the color of the coat, so this will give us like 3:1 ratio, right? The classical Mendelian ratio. However, this gene, as I mentioned, this gene can also determine whether a mouse is alive or not, right? So in reality, you can never see this genotype. Why? Because this genotype is lethal. So if a mouse has a genotype like capital Y, so this mouse cannot be born alive. So this is the situation. So now when you look at here, so the capital Y becomes a recessive lethal, because when we look at the heterozygous, this is a live mouse, right? This is alive and this is alive. So if we look at if we look at the second phenotype, so this Y is called the recessive lethal. This cannot survive. All right, what's the consequences? So if you look at a cross, so you only see like 2:1 ratio. Oops, 2:1 ratio. This is not a classical one gene Mendelian model, right? Because of what? Because we can never see this kind of a living mouse with this genotype. This is lethal allele. All right. So now here we talk about two concepts, right? One is the pleotropy. So this gene can determine two phenotypes. And if there's a recessive lethal, so you're not gonna see this genotype in the population. So this will give you slightly different ratios or proportions when you compare to the classical Mendelian one gene model. All right, so this is another example. So in human, the achondroplasia. So this syndrome actually is an example of the recessive lethal allele, because this genotype is not viable. So people, any individual, any human carrying this gene cannot survive. So you're not gonna see this. You only see this to genotypes in the population. And this is the allele, and it is a dominant. So if you see the heterozygous, so you're gonna see the dwarf phenotypes, and the wild type, the normal stature is carrying this kind of genotype, the homozygous recessive. All right, this is the same. So in cat, there's one gene that determines the shape of tail. So the homozygous recessive little t will give the normal cats with tails, and the heterozygous like this, this cat will lack tails. But you never see this kind of genotype in cats because they are not viable. All right. So as I mentioned, so usually the lethal alleles exist in this format, in this form, the recessive lethal. There could be some dominant lethal, but so it's very hard for this kind of allele to remain in the population because it is very rare. All right. Okay. Now, the fourth type is the codominance. So this is the fourth type of the relationship between multiple alleles for a single gene. We have talked about the molecular markers, right? DNA markers, DNA sequences. Usually they are codominants because in any heterozygous individuals, you can detect both alleles simultaneously. And in terms of the inheritance pattern, so the inheritance pattern for the codominance alleles, they are identical to the inheritance pattern of the incomplete dominance. So we mentioned this before. All right. So this is the example of the codominance. Okay, so so this is the gene that determines the human blood type. Okay, so this gene this gene has like three alleles. So let's say the IA is the first allele, and IB is the second allele, and I is the third allele. All right. So so either the IA or IB will be the dominant to the little I. However, if an individual has genotype like this, the IA and IB, so this in this individual, you're gonna see both alleles are expressed. Then we say, okay, so the relationship between IA and IB will be codominant because you can see both alleles are expressed, right? So the relationship between two alleles will change. So in this case, as I mentioned, if you look at IA with I, so their relationship is dominant and recessive, the same for IB and I, right? But if we look at the relationship between IA and IB, they are equal or they are codominant because the individual carrying this genotype will express both alleles. Right. Yeah, this is the details about this situation. So yeah, you can study this by yourself. And all right, so even we have more than two alleles, right? So when we study the classical Mendelian experiments, so we only talk about like maybe one gene, two allele, round shape, right? Wrinkled shape, and we can use the 2x2 Punnett square to help us to dissect the cross any monohybrid cross, right? Okay, then it will give you this, And this. Okay, so this will give you 3-to-1 ratio. And the genotypic ratio is 1-to-2-to-1, right? So when we talk about like more than 2 alleles, so we still can use, we can still leverage the 2x2 Punnett square to help us to study any cross. So suppose if we cross this two, so if this, so yes, so if these two people married, so these two people have a genotype like this, so we can still use the Punnett square to help us to study this mating. So for this parent, it can produce either IA or IB gamete, right? For this parent, it can produce either IA or little I allele. So for the progeny, for their kids, so it could be this genotype, IA, I, and IA, IB, and IB, I, right? Oops. Okay. Now, okay, so there are like how many genotypes? Actually, there are 4 genotypes here because there are 3 alleles, right? But if you look at the phenotypic ratio, so this 2 groups will be the blood type A, right? And this will be AB type, and this will be the B type. So you're gonna see the ratio like this. So it's different from the classical 1 gene monohybrid cross, but still we can use this Punnett square to help us to solve any genetic problem. Right? Okay. So this is another example in human. Sometimes when we talk about the relationship between different alleles for a single gene, now we still talk about 1 gene, right? Because later we're going to learn the relationship between 2 genes. So that's why I keep emphasizing now we are focused on 1 gene, but we want to see the relationship between different alleles for this gene. All right, the type of dominance between different alleles may depend on the phenotype studied. Okay, so in this example, okay, there's 1 gene called the HB in human, and the HBA is the allele, the wild type allele, and this so the homozygous, this allele will it's normal, is the standard genotype, and it will produce the red blood cell having the donut shape like this. Yeah, it looks like donut. Delicious. No, only vampire can say this is delicious. I'm kidding. All right. So this is the wild type allele. Okay, and this homozygous individual will have the normal standard red blood cell shape, donut shape. Okay, all right. And there's another mutant allele, so HBS. So that's the mutant, and this homozygous mutant allele will give a person a sickle cell anemia. So this is the shape. Okay. All right. Now let's look at the heterozygous. So in this heterozygous, so this will be normal. So this can produce the donut shape red blood cell only if this is in a standard situation. However, the same genotype, the heterozygous, will produce sickle blood cell if the oxygen level is very low. So for example, if you go to a high mountain So where the oxygen concentrate is very low. So in that situation, this genotype heterozygous will produce different phenotypes. So what does that mean? That means like the relationship between this wild type allele and this mutant allele, their relationship may be changed. So which one is dominant, which one is recessive? This depends on the environment. In the standard normal environment, like everywhere, so this is greater than this. This is dominant and this is recessive, right? However, if we change to another environment, like the low, very low concentration of the oxygen, so this relationship will be totally reversed. So this allele will become like dominant. So, yeah, so that's why, so we have learned this before, right? So the phenotype usually is determined by the genotype in the standard situation environment. However, if you put the individuals in the study under the different environment, this may change. This situation may change. So that's why the phenotype is not only determined by genetics; it's determined by the joint effect from genetics and the environment. So we need to consider the interaction between the genes and the environmental factors, right? So this is a very good example to show this. And also, so now here, if we want to study these two alleles by using the gel, we use the gel, ran the gel, okay. And so in this case, if we study these two alleles by running a gel, so the relationship between these two alleles will be codominant. Why? Because when we look at the heterozygous individual, we're going to see both alleles are expressed. So the heterozygous will show different phenotypes than homozygous 1 and homozygous 2. So the homozygous 1 and homozygous 2 have two different phenotypes. But if you look at the heterozygous, so you're going to see the third type of phenotypes, right? You're going to show both. So in this case, in this situation, so when we use different assay to study these two alleles, the relationship between these two alleles changed again. So now the relationship between these two alleles becomes codominance. Not like this one, right? This one is like, one is dominant, the other one is a recessive, depending on the different environment. Okay. So for the same allele, like HB, oops, A and HB S. Okay. So this is another very interesting example. So okay, so we have learned like, okay, for the anemia, so the wild type is dominant in the normal situation. However, it will change. It will change when the environment changes, right? So, okay, now when we look at this disease, so this situation, I'm sorry, the malaria. So this is a disease. So so let's look at the resistance level to this disease, malaria. All right, we have three genotypes here, right? So we have homozygous HBA and HBA. We have homozygous HBS and HBS. And we have the heterozygous HBA and HBS. All right, so when we look at these three genotypes in terms of the resistance to the malaria, so the heterozygous is the best one. So this is very interesting. So heterozygous is the best one. And in this situation, we say heterozygous is an overdominance, or heterozygous shows the heterozygote advantage. All right, in terms of this phenotype, this is the best one. The heterozygous is very interesting. And in crops, so probably in the future when you study crops, the plants, So sometimes the heterozygous is much better than their parents, pure line parents, in terms of some phenotypes. So in this case, we say that we gave a name to that situation is the heterosis, which means like heterozygous is better than the homozygous, right? So this is an example in human. Okay, now the fifth type, relationship between different alleles. This type is called the dominant and active alleles, or the poisonous alleles. So what does this mean? So let's look at here. So this is the first genotype, homozygous wild type. Homozygous wild type. So each allele can produce a functional protein, and these two functional protein can form a dimer, and the dimer like this is good, it's functional. So this will give you the wild type. Okay, however, if both alleles are mutant, so as you can see, you don't see any protein being produced. So there's no functional dimer here. So this will give you mutant type. Okay, for this one. Now let's look at the heterozygous. The heterozygous, you only have one dose of the functional protein, but you have like you also have like a non-functional protein. So if these two, the functional and non-functional, come together, so it cannot produce the functional dimer like this. So this one is also not functional. This will also gives you the mutant type. All right, which means, so if you look at here, so which one is dominant? Of course, the mutant is dominant, right? Because the mutant, the homozygous mutant gives the same phenotype as the heterozygous. So the mutant is the dominant. That's why we say dominant. But this dominant actually is not good. This dominant mutant allele will lead to a negative situation, or will lead to the loss of function, or we also say this is poisonous dominant mutant. So this is the mechanism behind this dominant negative alleles. Okay, so this is example. So some details. I don't want to go into this, because after the class you can study these slides. This just gives you a summary about this dominant and active alleles here. All right, the last type of the relationship between different alleles for a single gene is the multiple alleles. So now, so we have already talked about this situation, right? In human, so in human, there's one gene. So this gene actually this gene has 3 alleles, not 2. And the ABO blood group determine genes, there are 3 alleles. So this is very common actually in many other organisms, and in terms of other phenotypes. For one gene, this gene can take like more than 2 forms. For example, this is one gene in rabbit, and this gene can take different forms here, and different forms can produce different fur color, and those are the relationship between this. Like, let's say there are 4 alleles here, right? Those are the this represented the relationship between these alleles. All right, because there are more than 2 alleles, so as you can see, the phenotypes will be very complicated. So if we look at different combination of this alleles here, so this will give you different situations. So you can study this later in details. All right, so for example, so if this is the okay. Okay. Okay, so for example, this one, if there's one allele like this, so all this will show this phenotype. So because this one is greater than this one is greater than this one, and this one is greater than this one. All right. So I'm not gonna go details into this, but you can study this by yourself. So that shows how complicated if there are more than 2 alleles for a single gene. Okay, again, so here I would like to also emphasize this. Phenotype is not simply determined by genotype, genetics. It is the consequence from the interaction between genetics and the environment. So sometimes if you change the environment, probably you can change the consequence of genetics. So for example, this is a case. So this is a rabbit showing the white fur color. However, so in the standard situation, so this genetics will gives us like this phenotype, right? The white fur color. However, if we put a ice pack on the back of this rabbit, so it can we can purposely produce some like dark fur area on this rabbit. So this experiment shows, the phenotypes is usually determined by the interaction between genetics and the environment factors. Okay, so I think, oh yeah, this is the last slides for this part. Okay, so when we talk about the Drosophila, the fruit fly, yeah, as you can see, so we have learned this before, right? There's one gene on X chromosome, that's X-linked gene, and that gene can determine the color of the eyes in Drosophila. So in the past, for simplicity, we only talk about 2 alleles, probably. I don't remember W or small w, but in the nature, actually, there are many alleles. So there are more than 100 alleles, which means like, so probably there are like more than 100 different colors, eye colors in fruit flies. So this will increase the complexity levels in this phenotypes, right? Also, this will give a very, very large variability in this phenotype in the population, which is good. All right, so I think I'm done with this part. So again, so we just finished talking about the relationship between different alleles for a single gene. Now we're going to talk about different stuff. So we're going to talk about, the interaction between 2 or more loci. So from one gene, now we talk about 2 genes. So we're going to see how 2 different genes can interact with each other to determine some trait of interest, right? Okay, first, so this is what we have learned before, the classical Mendelian inheritance. So this ratio is for a single gene, right? So we have talked about this. So if we focus on just one gene, a single phenotype, so this gives us a classic 3-to-1 ratio for the monohybrid cross. All right, now if we study 2 genes, and if these 2 genes are independent, again, independent, what does independent mean? Independent means like they are on 2 different chromosomes, and they are doing 2 different things. There's no interaction between these 2 genes. Then, in this case, when we do the dihybrid cross, we will end up with this classical ratio, right? Mendelian ratio, 9:3 to 3:1 ratio, right? And this is the test cross ratio. So we're expert now, right? So this is, yeah, just a little piece of cake for us. All right. Okay. So now let's look at two genes. So if we have two genes here, if we do the dihybrid cross, and the results from this cross is showing like this, 9:3 to 3:1 ratio, then this is an indication that there's no gene interaction between these two genes. All right, so in this example, let's use snakes as an example. There are two genes here, all right? There's one gene, it's called the O. The O plus means the wild type, and the O is the mutant, all right? And the B plus is the wild type, and the B is the mutant. And these two genes are independent, and they are doing two totally different things. All right. So the O gene here, orange gene. So the wild type allele will produce the orange color, the orange pigment. All right. Yeah, on top of on the skin of the snake, and the black, the B gene. The wild type B gene will produce the black pigment on the skin, all right? They're doing totally independent things. So if both genes are expressed, if both wild type genes are expressed, you're gonna see both colors, orange plus black. So this is the this is the most commonly seen phenotype. It is called the camouflaged, right? However, if only one gene expressed, for example, so you only see the orange color, or you only see the black color. So the ratio is 9:3:3, and this is 1. So we all know why this is 1, right? So why this snake look like this? Because this is actually, if both genes are mutant like this, so this is actually gives the albino colorless, no color on the skin. So why we see some color here, pink? Actually, this is not the skin color, because the skin is transparent. You're gonna see the blood color, so you're gonna see through. So the color showing here is actually the color of the blood, right? So and excuse me. All right. So let's look at the biology behind this. All right, let's look at these two genes. So the wild type B genes will gives us the black pigment for the snake. So this is a biological pathway. And okay, so this B plus gene will produce some functional enzyme. And this enzyme is gonna work on some colorless precursors. So this precursors are colorless, and if there is a functional protein in this individual, the wild type, so this enzyme is gonna work on this precursor, and is gonna produce some black pigment. So the end product from this biological pathway is the black pigment. There's another biological pathway. So some different precursor still like colorless. If there's a wild type O allele in this individual, so this gene will produce some functional enzyme which is working on the precursor and will transform this colorless precursor into orange pigment. All right. However, this two pathway is totally independent, two different pathway. So that's why there's no interaction between these two genes. That's why you're going to see the classical 9:3:3:1 ratio. So this ratio gives us a genetic diagnosis. So in this case, we can say these two genes are independent. There's no gene interaction between them. All right. Okay, now let's look at some variants. So we always start from this ratio, but if there is some interaction or relationship between these two genes, you're going to see some variant ratios from this classical ratio. So the first one is the complementary gene action. So the two genes are related to each other, And this relationship is called the complementary gene action. And if this is the case, it will gives us the phenotypic ratio like this, 9:7. So how we related to this ratio to this ratio? So it's simple. If this, oops. So if these three categories showing the same phenotype, then we can group them together, and this will lead to the 9:7 ratio, right? Okay, let's look at the example here. Okay, so this is example. Suppose we have a flower, and the wild type is the blue color. Wild type is the blue color, and for some reason, like there's some mutant. So the white colored flower is the mutant. So we have 3 mutant lines. We use different the money sign to represent 3 different mutant lines: the U.S. dollar, the British pound, and the Chinese yuan. Okay, this represents 3 mutant lines, and we cross each of these 3 mutant line with the wild type, and as we expected, so in F1, we see all the F1 progeny showing the blue color. All right, which means the wild type allele is the dominant, right? It's greater than the mutant allele, right? However, when we do the F1 intercross, we end up with totally different results. So if we do the intercross for the first situation, so we have like 3:1 ratio of the progeny, F2 progeny. It's the same for the second situation. However, if we do the same thing, intercross for the F1 progeny, for the third case. Oh, sorry. Yeah. So my apologies. So yeah, if we do the intercross for the F1 progeny for these 3 situations, they all end up with the same results. Okay, the 3:1 ratio, right? Okay, now, so we would ask a question to ourselves. So, are all these white mutation the same? So when we talk about the 3 different mutations, are all the 3 white mutations are the same? Are they representing 3 alleles of 1 gene or 3 different genes? So to answer this question, we can use this so-called the complementation test. All right. So before we talk about this complementation test, let me tell you about the real biology behind. All right. So this is the biology behind, okay? The wild type, which shows the blue color in the flower, right? It actually, this blue color, this blue pigment in the flower petal is produced through this biological pathway. So first, yeah, there's a like colorless. So the precursor one is doesn't have any color. So colorless precursor one. So there's one gene called the W1. So if the wild type, this wild type gene will produce some functional enzyme, the enzyme 1, and this enzyme will work on this precursor and transform this precursor 1 into precursor 2. However, precursor 2 is also colorless. There's no color. All right. Now, if in this individual flower, if there's a wild type gene 2, which is the W2 plus, if this is a wild type, it can produce the functional enzyme number 2. And this enzyme number 2 is going to be working on this precursor 2 and transform this colorless precursor 2 into the blue product, the pigment. All right. So if this pathway is functional, then you're going to see the wild type. Okay, now look at here. So this is the wild type. We have 2 genes, right? And each gene has two copies. So in the wild type, so both genes have the wild type alleles, W1 and the W2. All right. Okay, so this is the wild type. Now this represents 3 different mutant lines. So for the first one, there's a mutation occurring in this gene. And in the second one, there's another mutation occurring in the same gene, but in different location. So look at here. So if you compare this one with this one, the mutation occurring in the same gene, but in different location, right? So let's look at the third one. So this is different. For this mutation, actually there's a mutation occurring on the second gene, the W2 gene. And the W1 gene is still wild type. Okay, so this is the biology behind. Okay, now if we cross the first one with the second one, the mutant, what happens here? So this is the result. This is the progeny from this cross, right? So why this is still mutant? Because so look at this the 2 alleles for this gene. So they are all mutant. So this pathway is still blocked because of this situation, right? Okay, even if this gene is wild type, but because this gene is mutant, so the biological pathway is still blocked. This will gives us the same mutant phenotypes. However, if we cross the second one with the third one, so let's see what happened here. All right, this for this mutant, this gene is mutant, but in this individual, this gene is wild type. So when they mate, you're gonna end up with the heterozygous. So one copy is wild type and the other copy is the mutant. The same thing for the second gene. All right. So if you cross this individual, you're gonna end up with the heterozygous. One copy is the wild type and the other is the mutant. So which means like both steps in this biological pathway is functional, then this will give us the wild type, the purple flower. So why is this? Let me erase this. Because in this cross, the 2 mutant can complement each other, because in the original mutation lines, the mutation occurs in 2 different genes. So that's why if you cross these 2 mutant lines, so these 2 mutant lines can complement each other. So we say this is the complementation test, right? So this is the the functional pathway showing the biology behind these 2 cross, right? All right, because the first cross 1 and 2 end up with this. So this is still mutant. So the first step is still blocked. So you're not gonna see the purple end product. However, if we cross number 2 to number 3, and both genes are heterozygous, which means like both steps are functional in this pathway, and we're gonna see the wild type phenotype. Any questions? Okay, good. All right, so now let's look at the ratios. All right, so the same model, if we cross the first a mutant with this mutant and end up with the purple flower, which is the double heterozygous. And if we do the intercross between this F1 progeny, we're gonna so if we just look at the genotype, so this will gives us like 9 portion of this, 3 portion of this, 3 portion of this, and 1 portion of this, right? However, because if we want to see the wild type phenotype, we need to see both genes are functioning. So this is the only case that both genes will be functioning. and this will show the wild type phenotype, right? If we look at it here, only the first gene is functioning, but the second gene is mutant. So the second step in the pathway has been blocked. So this will give us the mutant phenotype. The same thing for this one, right? Only the second gene is functional. And for this one, both genes are non-functional. So this 1, 2, 3 categories actually yield the same phenotype, the mutant phenotype. So we can actually group them together, and this will end up with this ratio. Yeah, is that clear? Okay, very good. All right, so we just used one example to show this 9:7 ratio. And this 9:7 ratio can also arise from the interaction between. So here we use a pathway, right? This is a pathway example. However, this ratio can also arise from the interaction between a gene and a regulatory protein required for the expression of this gene. All right. So let's look at this example. So in a normal situation, okay, so this is the gene for the regulatory protein. Yeah, if this is a wild type allele, it can produce some functional. It can be expressed, transcribed, and this transcripts can produce some functional protein. And this is the regulatory protein. This protein is gonna bind on this region for the gene A, and this region probably like the regulatory region or promoter region. So only if this protein binds to that region, so this gene can be transcribed and can be used to produce the functional protein. And this functional protein can lead to the wild type phenotype. All right. So this represents the 9. Okay, this category. All right, now look at one situation. So if the regulatory gene has something wrong, so the mutation occurs here. So this is the R, not the R plus, and it cannot produce. It cannot be correctly transcribed, and the transcripts cannot produce any functional protein. So this protein is not useful, and it cannot regulate the expression of this gene, and this gene cannot be transcribed. And as you can see, you do not see the protein, functional protein.
10%.
Hello, do you mind if you can mute yourself? Yeah, we can hear some like background noise. Oh, thank you. Thank you. All right. So this will gives us the mutant type. The same thing here. Let me stop for a little bit. Okay. Okay. Yeah, it's better now. Okay. So as I mentioned, this is the 9, this is 3, and this is another 3 because of this gene is a mutant, so it cannot produce any functional protein for the wild type, right? And this is of course the one portion, and because these 3 categories shows the same phenotype, so we can group them together, and this is the 7, and this is the 9. So, all right. Okay, so this table shows you an example. So in the exam, I could give you some question like this. So we have like 4 mutant lines here. 1, 2, 4. 1, 2, 3, 4. So they are mutant line. If they cross by itself, so you're gonna see the mutant type, right? Mutant type here. However, if we cross 1 with 2, so we're gonna see the wild type, which means, okay, so this mutant and this mutant can complement each other, right? So if we do the complementation test, so this mutant can complement this one. The same for this one. So 1 and 3 can complement. So if you cross this two, it will produce the wild type progeny. The same thing here. However, if you cross number 2 with number 3, so you do not produce any wild type progeny. So which means these two mutant lines cannot complement each other. So if I ask you a question, so in the final exam, because of the time issues, I cannot give you like the questions in a short answer format. I'm going to give you multiple choices like this. So this could be a example of the question. So if I gave you the results like this, so from A, B, C, just choose the right one. So in this one, so of course, based on the data, we can say the mutation affect the different genes, except that the number 2 and number 3, they affect. So in these two mutant lines, the mutation affected the same gene, right? That's why they cannot complement each other if you do the complementation test using these two mutant lines. All right. Okay, so in the neurospora, so we can also do this kind of complementation test. So we have like mutant 1, mutant 2, and mutant 3. If we cross mutant 1 with mutant 2, so if we generate this results, that means like these two complement each other, right? So the same for the mutant 2 and mutant 3. If we generate something like this, that's the indication of the complementation. So when we cross the mutant 1 and mutant 3, so if we do not recover any wild type, then this two mutant showing the mutation in the same gene, right? So this is the biology behind. So I'm going to leave this to you. So you can, after class, you can study these two possible crosses and to understand why the number 1 and number 2 and number 2 and number 3 can complement each other, but not for the mutant 1 and mutant 3. All right. Okay, so we can use something called, so in the neurospora, the complementation can also be tested using the heterokaryon. So for example, this is the mutant. It cannot grow on the it cannot grow without the arginine. So the same for this types of mutant. It cannot grow without arginine. However, if we fuse these two mutant lines, if we can get the fusion of these two mutant lines, so the fusion of these two mutant lines can grow by itself without adding any arginine into the plate. It can grow by itself. So this is another experiment that we can do for the complementation test. All right. And so I think, yeah, we have time to talk about one more situation. So we just finished about the complementary gene action. So this one is, I put this one in the first, right? The number 1, so, which means this is very important. This one is very important. So I hope everyone understand this. And definitely, yeah, you're gonna see some questions related to this in your future quiz or in the next middle term. Okay. The second one is the duplication of gene loci or duplication dominant epistasis. Right. So what is this? First, So this is another variant from this ratio, the classical ratio, right? So if we got this ratio, it's easy for us to understand how this ratio is related to this ratio, right? So this is because if these three categories show the same phenotype, then we can group them together, and this will yield 15:1 ratio. So usually, in this case, we're gonna see the redundant gene function for these two genes. So there are two genes. There are two genes in this individual, in this organism. However, these two genes are doing the same thing. They are doing the same thing. Yeah. So that's why you're gonna see this kind of ratio. So why is that? So for example, if we have two genes, one is A and the other one is B, if the wild type can produce, if the wild type allele for either of this gene as it exists, it can produce the wild type phenotype. So the same for this one, right? Even for the second gene, it is mutant. However, for the first gene, it is wild type. So it can also produce wild type for this phenotype. The same thing for this category, because A gene and B gene are doing the same thing, identical thing, same function. That's why we can group these three, and this will give us 15:1 ratio. Only this one is the mutant. Let me see, what time is it now? Also, maybe we can talk about one more here. So all right. So the third one is the recessive epistasis. All right, this gives us another ratio here, 9:3:4. So it's easy to understand, right? So this is, so they all came from this ratio. And so how can we relate this ratio to this? So if these two categories showing the same phenotype, then we can group this two category together to have 4, right? So this is the ratio. How we related this classical ratio to this new ratio to represent this relationship between these two genes, recessive epistasis. All right. So this is the pathway. So behind the biology behind this this type of relationship between these two genes, which is the recessive epistasis. Okay, let's look at this pathway. So this is the pathway. So the end product in a wild type individual, wild type phenotype is the blue flower, the blue pigment in the flower petal. All right, if everything is fine, the wild type, so we have one gene here and one gene here. So this gene can produce some functional enzyme, and the gene M can also produce some functional enzyme. Let's look at the function of the first gene. Okay, the first gene will work on some colorless precursor and transform this colorless precursor into some red stuff, intermediate product. Okay, so this is the intermediate product, and this one, if the second gene, M gene, is also functional, and this gene can work on this intermediate product and transform this pink thing into the blue pigment, the end product. So this is the normal standard. All right, that's why if both genes are wild type, you're going to see the blue color, right? This is a wild type. However, if the first gene is okay, so we can have the pink intermediate product, right? But the second gene is mutant, so this pathway is half blocked. So this is blocked here. So then we only see this color pigment in the flower petal, right? So this will give us this phenotype. Right? Let me erase this. Okay, so this is the wild type, and this is because of the second gene is mutant. Now let's look at the first gene mutant. So if the first gene is mutant, All right, so this pathway has been blocked, so it cannot reach this step. So this in this individual, so no pink product can be produced. So no matter no matter what the second gene is, either the wild type or mutant, it cannot be functional, right? Because there's no such substrate that can be used can be used by this gene. So if the first gene has something wrong, the mutant, you're gonna see the same phenotypes here, white phenotype. There's nothing can be produced. You only see the colorless, the white flower. So which means we can group these two categories together, and this will get us the 9:3:4 ratio. And this is the pathway for this example. All right, I think, yeah, I'm gonna stop here because we do not have time to finish all, but yeah, we're gonna continue this next time. So any questions for today's slides? Is that clear? Yeah, okay, so tomorrow's quiz, I don't think I'm gonna cover today's stuff. So tomorrow's quiz will be focusing on the neurospora. So how we map the genes between genes or between gene and the centromere using the haploid organism model neurospora. So that's for tomorrow's quiz. For next quiz, probably, yeah, I'm gonna include the stuff we have learned today, either the interaction between different alleles within one gene, or the interaction between two different genes. So that's for the next week. For this week, tomorrow's quiz, yeah, we're gonna focus on the lecture 9, I think. Today is lecture 10, right? So lecture 9 is about like the neurospora mapping. Yeah, sure.
Hi, Professor. I actually had a question on slide 30 about the lethal alleles.
Yes, 30. Let me see. Okay.
So I was just confused as to why it's capital Y, but then it's recessive lethal.
Okay, yeah, this is indeed confusing. So here, when we talk about the lethal allele, so I introduce another term, right? The pleotropy. Yeah, this means like a same gene, let's say the Y, big Y and small y, can determine two phenotypes here, right? So let's see. So the first phenotype this gene can determine is the fur color, right? The coat color. The coat color. And the second phenotype this gene can determine is whether alive or not. This is related to the lethal, right? Or not. Okay. So we put capital Y and the small y here. So this notation is only related to the first phenotype. So why is that? Because so let's leave the lethal stuff alone. So just look at the color of the color of the coat. All right, if everything is here, right? So as we can see, this will gives us the yellow coat color, and this will gives us the black. So how do we determine the relationship between big Y and small y here? So in terms of this phenotype, we can say the big Y, capital Y, is greater dominant than to the little y, because the heterozygous is showing the same phenotype with this. Is that correct? Is that correct? So because the heterozygous individual showing the same phenotype with the homozygous this, so we can say this big Y is dominant to the little y. So remember here, we only talk about the first phenotype. Okay, let's erase this. Now let's look at the second phenotype. Second phenotype. Now this is the phenotype whether a life can survive or not versus not. So this one cannot survive, but this two can survive, right? Do you agree?
Yeah.
And if you look at this phenotype, so the heterozygous has the same phenotype with this homozygous. So which one is dominant in terms of the second phenotype? The little y is dominant than the big Y. Do you agree?
Okay, that makes a lot more sense.
Yeah, so that's why we say, okay, this is a recessive lethal because they are recessive in terms of the being alive or not. Yeah.
Okay, and then for dominant lethal, so would it be rare because if it were dominant,
It's true, yeah.
the
so
progeny would just die right away and then it wouldn't be passed down as well?
That's right, yeah, that's exactly like that. So that's why, so usually when we
Okay.
talk about the lethal, so this is the most common situation talking about recessive lethal.
Okay, gotcha. Thank you so much.
Sure, sure. Any other questions? Okay, so if you do not have any questions, so we're gonna stop here. And again, tomorrow, so the quiz will focus on the neurospora mapping, and today's stuff, like the relationship between different alleles within one gene, or the relationship between multiple genes. This will be on your on the quiz of next week. Okay. All right. Let's stop here. Thank you, everyone. See you on Thursday. Thank you.